Once again, I am welcoming you all in this third session of this first short-term training program under AICTE AQIS on image processing and its application. For this session, we have with us Dr. Abhimanyu Singh Garwal. Dr. Abhimanyu Singh Garwal is a postdoctoral scientist in AG Research, New Zealand. He finished his Bachelor of Engineering in Information Technology from University of Rajasthan in 2006. He received his Masters of Business Administration in Information Technology, followed by Master of Technology in Information Technology from Rajasthan Technical University, Kota, respectively in 2010 and 2012. Abhimanyu Singh received his PhD in Computer and Information Science in the area of bioinformatic inspired images analysis from Auckland University of Technology, New Zealand in 2018. His first postdoc was at Massey University, New Zealand, working on hyperspectral imaging for fooding grading, in particular potato zebra chip disease detection. He developed models for real-time segregation of healthy and zebra chip infected tubers. He also used hyperspectral imaging for the identification of adulteration in Manuka honey. I welcome you, sir, and now I want to hand over the session to you. Abhimanyu, sir.
Okay, so thank you for that. Thank you for my introduction. So he explained everything. Now I'm just sharing a little bit more about me. So basically, I belong from Jaipur, and already he explained my education and experience. So I'm not going in that area. I'm just explaining the research area in which I worked. So digital watermarking, hyperspectral image analysis, bioinformatics inspired image analysis, speaker identification, currency security, and database optimization. Today's topic, as all of you know, application in digital image processing and focused on here on hyperspectral imaging. So first I will explain the application of digital image processing, and then I will explain about hyperspectral imaging. So everybody knows, like, it's a very simple application of image processing. Image processing is image sharpening and restoration. So what we do in that, we manipulate the captured images in a way to achieve desired result. Like, this is the original image. You can blur it. That's the image processing, image sharpening, restoration. You can zoom it. It's a simple image sharpening and restoring operation. The next application in the medical field, like gamma ray imaging, PET scans, X-ray imaging, medical CT scan, where you can capture the image from different resources as per the requirement of the medical analysis. When you capture the image, after that, you have to apply image processing techniques to get the required information. Like in X-ray, you need to analyze the bones are fractured or they are not. In another CT scan, you have to analyze if there is a cancer or not, is a healthy tissue or not, something like that. Next application is remote sensing. Remote sensing is a very interesting area where the area of Earth is scanned by a satellite from a very high ground, and then it is analyzed to obtain information about it. So you can see in the picture, like almost 770 kilometers, and is moving around 24,000 kilometers per hour, is capturing the image at a very fast rate. And as a result, we will get some ground information, and then we apply some image processing technique to get the information about the field area, like where is the potato, maize, barley, oilseed rape, fly, wheat, sugar beets, so with different color schemes. Another interesting is the machine robot vision. So if you see this video, So basically what this robot is doing is identifying this hurdle, and then by an image, and then in a background some image processing algorithm, artificial intelligence algorithm run, and according to the object height and length, it will direct him to jump or take the next step. Like he jumped in this way. Now he found another obstacle. So another image capturing, and after that image capturing, the same process repeated, and he took the action. Then the next image, then the next image. So it make robot to see things, identify them, and according to them, they will take the next step, what they want to do. And another example of this, like here you can see there are a lot of obstacles, but by artificial intelligence algorithm, he is able to identify the path through which he want to travel, and he is traveling very good. Like here you can see it hit by some obstacle, and he changed his path quickly. Now the biggest challenge is to capture the images in the night mode, but there is a very good algorithm nowadays that they can detect very well in the night mode as well. So that's the very interesting application for the image processing. Next application of image processing is pattern recognition. In pattern recognition, by using definite kind of image processing, what we do, we can identify the different kind of objects in the images, like if in an object like handwriting, or if in a on an image there is a ball or there is any obstacle, something like that. So we have to look for that pattern, and we can train the system or model so to identify the pattern, and then on the basis of that pattern, that model will predict this is a ball, or this is a tennis ball, or this is a cricket ball, something like that. So I did some work on this pattern recognition system. So this is my PhD work where I converted images into DNA letters like adenine, thymine, guanine, cytosine. And then from that DNA encoded images, I extracted common patterns. And that common patterns are used to generate phylogenetic trees to group images in their respective categories. So for extracting patterns, like I used some sequence alignments. That these are the bioinformatics algorithms, sequence alignment algorithms. And what we did, like we took some watermarked images and we degraded them by using print and scat methodology to see if this method is able to detect the degraded and non-degraded image on the basis of watermark and non-watermarks. And we successfully, with 100% accuracy, we are able to generate a phylogenetic tree that can segregate watermarked and non-watermarked image by using this approach. The next stage of this research is bioinformatics and inspired image identification approach. So again, the images were converted into DNA letters, and then again the common patterns were extracted from biologically based DNA converted images, and we call them as a signatures. And these signatures are used to identify the image with watermark and non-watermark images. It means, like, if an image belongs to a person and it's like a fingerprint of that person, so we can extract the signature from the image photographs and from the image of that person. And then if we want to identify, yes, this image belongs to Abhimanyu, or this image belongs to Rakesh, or this image belongs to Manish, whatever. So that's the application of this approach. Now, my next pattern identification image processing is an empirical approach for currency identification. So where what we did, we extracted a two-part feature vectors, where is color features and texture feature. Color features have matrices are kurtosis, central movement, mean, variance, standard deviation, and skew. And texture features that we extracted, there's a gray, like the entropy level calculated for 4 features from the gray level concurrence matrix, namely correlation, contrast, energy, and homogeneity. Finally, the banknote in question is classified by a feed-forward neural network, and then measurement of similarity between existing samples and suspected banknote is as a output. So this is the user interface that we developed for the New Zealand currency identification. So this is the test banknote, and when we input the test banknote, there is a similarity score. There is a back and front side. So we scanned and prepared a database of back and front side of the New Zealand dollar. And when we put in our system, scan and put in our system, then there is a similarity score that is 99.82%. So above 99%, we can say, yes, the banknote is authentic, and it will gives as an output this image. We input this, and from the database that we prepared, it will show us this node, this backside, what we scan belongs to this. So within 99 point some accuracy. Now the next is the hyperspectral imaging. That's a very interesting application. Normally, we deal with two-dimensional images. Two-dimensional images at one wavelength, like suppose two-dimensional images, there is an electromagnetic spectrum, and from 550 to 750 is a visible, and from 750 to above is infrared, and then near infrared, and all like this. So in hyperspectral images, images are captured, two-dimensional images are captured at different wavelengths. Like this, you can see the same car image is captured as lambda 0. That means any wavelength, it might be any wavelength, 750 or 650. Then another lambda 1, lambda 2, lambda 3, lambda 4, like this lambda n, as much as you want, like how many wavelength you want to capture, 200 wavelengths, 300 wavelengths. So these two-dimensional wavelength, two-dimension pictures at different wavelength contain different kind of information, and from that information you can extract the information as per your requirement. Suppose on the handle, on a door handle, there is some TNT explosive on the door handle. So from the analysis of the spectral analysis or spectral signature of the TNT, you can identify, yes, door has the some TNT on it. Another application of hyperspectral imaging Like this is the field area scan from the aeroplane, and which has some water, some field, some soil, some rocks. So suppose we capture 235 waveband. That means these are the pixels at different wavelengths for the atmosphere. These are the pixels of the soils at different wavelength. Again, these are the pixels for the water at different wavelength, and these are the pixel for the vegetation at different wavelengths. And you can see the spectral information of the atmosphere, soil, water, and the vegetation is completely different. So on the basis of spectra, you can easily segregate if this is a water, or this is a soil, or this is a vegetation. And normally it is used to identify the forest area, how much area is covered with the forest and how much area is the dry land. So when you capture the image, you can identify, yeah, there's that kind of the forest, or pine forest, or eucalyptus forest, what kind of forest there as well. It can also identify by using hyperspectral imaging. So that's the project on which I worked in Massey University. Hyperspectral imaging for identification of zebra chip disease in potatoes. So zebra chip disease in potatoes is a disease because of that. It causes stripes in the potatoes, and the chips become unacceptable to the customer. So hyperspectral imaging, so we captured potato images at different wavelengths. So we captured at different 235 wavelengths. That means we captured 235 two-dimensional images of the potato. So for understanding this, we have to understand what is spectroscopy. Spectroscopy is basically when a light fall on an object, some of the light is emitted, some of the light is reflected, and some of the light transmitted from the object. So whenever light interact with the material, so how it interact and what is the effect, that is called as spectroscopy. It examines the light behaviors in the target and recognize based on their different spectral signature. And a spectral signature can be identified from the spectrum of the material, and spectrum describe the amount of light in different wavelengths. It shows how much light is emitted, reflected, or transmitted from the target. So like from this graph, you can see this is the intensity, different intensity at different wavelengths. So suppose here is high, here is low. So that's the intensity versus wavelength. Now, again, the spectral signature is like the fingerprint of the human being. It's unique to the material. Like if it is a plastic has a different kind, has a particular fingerprint. That means it has a particular spectral signature. In the same way, leaves has their definite spectral signatures. Fingerprint. So spectral signature can be considered or equivalent to the fingerprint of the human being. Is unique to the human beings, like spectral signature is unique for the materials. Now, to capture this spectral signature, we need an instrument that is called a spectrometer, and spectrometer will capture the light, and that light is called as reflectance, because we always capture the reflected light, and that spectrum is reflectance spectrum. And the instrument that we use to capture this spectral information, that's called as hyperspectral camera. So hyperspectral camera scan the images to get the hyperspectral data, and from that we can identify what kind of material is it, and if there is a problem, and the location as well. So hyperspectral camera is capturing the spectral and spatial, both information we capture. So we can differentiate in between the hyperspectral and the RGB data. RGB data is captured at 3 wavelengths, red, green, blue. But hyperspectral data always captured at more than 200 wavelengths. So it's a book of image, like the same leaf is captured, like same leaf, two-dimensional images, 235 images are captured. That means at 235 different wavelengths. So there is a book, and that book is a of images of the same leaf, but this book doesn't contain any index. So if you want to know some information, like at which page, like in a book, you go to the index, or suppose you want to study about the hyperspectral imaging. So you will look the index of where is the hyperspectral imaging, you go to the directly to that page number. But in the same way here in hyperspectral data, we have a book, but book is black box because we don't know which page contain what kind of information. And if we need to extract that information, we want to extract that particular information. Suppose I want to extract zebra chip disease information. So I will go to the definite page number. I will develop an algorithm, and from that algorithm, I will find, oh, that's the page number. And from that, I can extract the information required for detecting zebra chip disease. So this I explained previously. So by hyperspectral imaging, we can analyze every pixel, like this pixel, where and what is it infected or is it healthy? So both information we can obtain from hyperspectral imaging, which location is infected, which location is healthy. So that's the advantage, like we have the spectral and the spatial information using hyperspectral imaging. Now, there are different kind of hyperspectral cameras, and they have different ranges as well. Some work in ultraviolet area, some works in visible light, some works in near infrared. It depends, and they are very application specific. That means 400 to 1000 nanometer depends on some application, 1000 to 2500 nanometer for another application, 400 to 2500 nanometer for another application. They are very specific to the application. Now the zebra chip disease, because of this zebra, all this study happened because this zebra chip disease causes millions of dollars of chip industry loss in New Zealand. So they want to reduce the potato chips wastage. For that, they created this project. So what is the zebra chip disease? This is a potato plant, and there is a psyllid larva containing Candidatus libribacter bacteria. Candidatus libribacter bacteria, and when it inject the stem of the plant, it larva contain that bacteria, and it goes down to the root of the potato, and it infect the potato from surroundings, from the periphery of the potato first, and then it contains the stripes like the zebra chip when you fry it. So we did some preliminary tests. So what we did, we firstly use near infrared spectroscopy, and we get a good detection rate, around 95% diseased potato detected as disease, and healthy in the validation set also 94% of potatoes, diseased potato will be detected as disease. And the data set is like around 600 potatoes for the calibration and 400 potatoes for the validation. After that, we tried to mimic the zebra chip disease by tempering the potato using needle to develop the zebra chip disease, zebra disease pattern, and we can see we can easily segregate the tempered and healthy potato with the 95% confidence level. Last test is just for detection of, hello?
Yes, sir.
Last test, yeah, just something, noise is coming, that's why I'm just, yeah, last test we did to identify that normally when we harvest the potato, they are dirty. So can we scan the dirty potatoes or washed potato? So we conducted another trial test where we captured the images from the dirty potatoes and the hyperspectral images of the dirty potato and hyperspectral images of the washed potato. And we can see there's a clear-cut difference in the, like this blue is the dirty potatoes, and this orange color is the healthy potatoes. So there is a clear-cut difference. So we decided that we will use for our big trial the washed potato. So then we conducted a very big trial, and that's the methodology for the trial. So we captured the images, then we calibrated the image, then we did the segmentation. We got the segmented image, then we extracted the individual potatoes from the tray, and then we extracted the region of interest, and then we did some spectral analysis. So we got a spectral data matrix, we applied some spectral data pre-processing, then we got the pre-processed spectra, then we did PLSD analysis, then we developed a optimal model, calibration model, and finally we found the which wavelength are most suitable for the model. And then we did another validation test, that is it diseased or healthy. So we cut it down in and then we see, oh, this is healthy, and there's the first validation, visual checking of our diseased potato. And the second validation, then we cut it down at 2 mm chips, and then we fried, so see they are really diseased or not. So that's the methodology. So that's the big trial. This container full of potatoes is empty half after half experiment done, and then I realized I have to take pictures. So it's too late. And then this is the hyperspectral imaging system, and this is the hyperspectral image of the potato. So there's the actual trial, some photographs. Now, when we capture the hyperspectral images at 235 wavelengths, so this is a video for showing how these, so yeah, display.
Sir, I have one question here.
Yep.
Sorry to interrupt you. Actually, these potatoes, can it be that half of the potato is infected and half is not infected?
Yes, this is a mixed data set. So we are mixed data set as a half. I have an answer for this question in my next slide. So how many potatoes are diseased and how many potatoes are healthy? So from these 8 potatoes, initially I don't know which one is diseased, which one is healthy. We are just capturing the images, and after that we cut it down to check which is diseased or which is healthy, and then we kept a record of that. This potato number 5 is diseased, potato number 6 is healthy, potato number 7 is diseased. So we kept the record of this. So it's around 3,352 potatoes are scanned, and from 3,352 potatoes, more than 50% potatoes are diseased. Initially, we don't know, but after trial, after scanning the images, we know which is disease and which is healthy. So it's a mix. It's not all healthy, all disease. You got your answer? Hello?
Yes, I got my answer.
Yeah, okay.
So one more question is that suppose if we are seeing at one potato, is it actually, I mean, would we be able to identify without cutting it whether it is?
Yes. That's the magic of this technology. Without cutting.
Okay.
Without cutting, that's the magic of this technology, that is a non-destructive technology. Non-destructive means without touching, non-touching, no cutting. Just you have to scan, run my algorithm, and then you can it will give the result, it's a disease or healthy. That's all, nothing.
Okay. Thank you.
Yeah. So these are the different, just for the explanation, different images. So that's the validation test, and that I explained you. We cut them down, fry them to check either they are diseased or healthy. So this is the extremely diseased potato, highly infected, mildly infected, and this is the healthy potato chips. So there is a publication on this. You can see on the online. I can share a link with everybody, so you can go through that, the paper, and then you get your almost all the answer. And if you have any question, you can ask me later as well. So yeah, I will come back to the presentation. 3352 potato tubers are scanned. Out of that, 1585 healthy tubers and 1767 are infected tubers. So more than 50% potato are diseased, and in a mix of potato, potatoes. It's not like the all healthy and all disease. So like this, so now I know this number 1 is disease, number 2 is healthy, number 3 is healthy, number 4 is disease, number 5 healthy, number 6 disease, 7 healthy, 8 healthy. And we use the same like numbering system. So we can easily track back which one is disease, which one is healthy. So now we have this spectral information and we need this information, that means results. So for this, from this step to this step, we need a methodology, and that's the methodology that we developed to get the results. So before we start working, we have to know the images and the test and what are the future issues before we start analysis. So there are 3 issues: spatial issue, spectral issue, and the selective information. So spatial issues like shadows of the potato, shapes, because potatoes are in different shapes. They are not in fixed shape, dead pixels. There are some unnecessary area, how you are developing the mask, etc. These are the spatial issues that we are going to address. Spectral issues, because when we are capturing the spectra, there are spikes because of the external lighting system, some baseline drift, some noise, instrumental noise, environmental noise, some strange wavelengths behavior, and the selective information in the spectra or in the surface. That is another issue. So image processing part, what we did. So this is the hyperspectral uncorrected image, because when we are capturing the reflectance, actually we are capturing the radiance. So we have to remove the dark and the white reference to get the calibrated image. You can see the difference after the calibration. And after the calibration, we have to remove these labels, this white strip, these black backgrounds. So we applied some segmentation algorithm using thresholding and morphological operation. We got the segmented image. After obtaining the segmented image, we extracted each potatoes with their numbers. So this is the individual potato extraction. And after that, we extracted a region of interest from the center of the potato, 20 by 20 pixels. That means 20 by 20 square for the center of interest. So we got a cube. Image cube means 20 by 20 from the centroid of the potato. So there are at these 2-dimensional spaces at 550, 570 nanometer, 580 nanometer. That means there are number of images behind that. So it's 235 images for one potato. After image processing, like this 235 wavelengths from 550 nanometer to 1700 nanometer. Then we use some spectral data processing. This part is for the spatial image processing, and that is the spectral part of image processing. We use first derivative, second derivative, standard normal variate. So we want to identify which one is the best approach for our case. So we applied all different kind of pre-processing, and we identified this is the best process. And after that pre-process spectra, we did analysis using PLSDA, and we tried different combinations. And from that different combination, we can see that it gives an accuracy of 92%. And after that, we try to identify optimal wavelengths, and we found, because initially we selected 235 wavelengths, important wavelengths, and from 235 important wavelengths, we want to identify which wavelengths are relevant for zebra chip disease detection. So we found 42 wavelengths are important for the zebra chip disease detections. And like the spectral range that we use is from 550 nanometer to 1700 nanometers. So the camera cost above 1000 nanometer is very high. So we want to see below 1000 nanometer, how many wavelengths are important? Is there is a change in accuracy or not? So we conducted another analysis and we found 34 important wavelengths are important for Jabra chip disease detection below 1000 nanometer. And it's almost the same accuracy, 92% accuracy on the validation model. And then we got the final prediction model. So that's the calibration process, 3352 potatoes. This is the data set calibration, 2,600 out of which 1,265 is healthy tubers and 1,415 are infected tubers. And after that, we did a model validation on 672 samples. That means 20% of the approx of the total sample, 320 healthy tubers and 352 infected tubers. So always it's a mix of more than 50% disease and healthy tubers. So the accuracy, the cross-validated calibration for diseased is 92.92%. For the validation is 92.3%. So almost is a very good accuracy. But there is a still false positive that disease treated as good. That's we have to improve. And we improved as well using convolution neural network and 3D CNN. But that's the next stage of this project that we did already. And that's in the process of writing paper. So now the next stage is like we went to a company. This the Vyama is a New Zealand-based company. So we discussed with them this approach, and they are very happy to develop a machine on this project. So whole system. The first step for this, we have to buy a hyperspectral camera. The second is light system, and then third is model transferability. The model we develop, how it can be compatible with the machine that we are developing. So hyperspectral camera, when you go to the market and you want to buy, you have a lot of option and you feel like this. Oh, which one I buy? There's a lot of cameras. But actual situation when you go to buy, because your requirements are very specific and you will feel like this. Which one? Don't know. So why this is? So this because these are the three kind of hyperspectral camera. The first one is snapshot. Snapshot, it will capture the whole image at once. Second one is the pixel by pixel. So it scan one pixel at a time. And third one is line scan. So line scan, it scan one line of pixel at one time. So we have to make a trade-off in three things. So again, I don't know. It depends on the application. So there are three things that decide which camera should I buy. Application target, the speed of capturing the image, and the price. But at last, what it get highlighted? The price always takes the most priority thing. So we have to become a trade-off, like which camera should buy and what price should be also reasonable. So it's extremely difficult to define a general framework on hyperspectral camera to buy, strongly dependent on the application, doing what in where. That means in what, in industry or in a lab. So it depends the outer environment condition, how dynamic the conditions are. Next is the LED lights. LED lights for lightening the system. So there are different lights. It depends what kind of light suitable for your object. So these are the different lights like ultraviolet range, visible, infrared. What kind of light you are using? If you are using infrared camera, so you need this infrared lightning system. If you are using just visible, so any of these light is okay. Ultraviolet, then you need a different kind of light. But we are working in near infrared, so I think that will be fine for us. Now prediction model. So hyperspectral imaging that we did our trial, so it can identify Jabra chip infected potatoes with 92% accuracy. And now how we can integrate this sensing system into protected data, that is Vyama system that we are doing. Thanks. Any questions?
It was a very nice session. Actually, I have a question regarding that. Actually, you have tested this application on the potato. So what are the other type of item on which this algorithm can also apply?
As a mix, this question is not framed very well. I will just correct it. Like you are asking two things. So you are asking first thing, your question is what kind of other application it can be used. Second question, the algorithm. So it can be used, I will give you a background for this, then you can understand. Initially, hyperspectral imaging was used in satellite in 1970s. That's the initialization of the hyperspectral imaging. But at that time, that hyperspectral cameras are very costly, very costly. So normal like industry cannot afford to apply in the real-time scenario. But with the passage of time, the hyperspectral camera become cheaper. And in the last 5-6 years, that hyperspectral camera has become more cheap. So it means we can we can afford these camera for daily life. Like if you want to detect a defective apple and a healthy apple, you can apply for this. If you want to detect defective almond, or if you want to detect a healthy almond, you can use for that. If you want to detect any defective, in a broad term, you can say you can detect, this technology can be used to segregate a healthy or diseased fruits or vegetable, whatever your application you want. And algorithms are very specific for the you can say for the fruit or for the vegetable, whatever fruit and vegetable you are choosing, you have to develop algorithm as per your requirement. You have to develop the algorithm. It's not universal algorithm. You can use one algorithm for the another scenario. You have to develop algorithm specific for that fruit, specific to the conditions, environmental conditions, and specific to the you can say fruits and vegetable as well. Yes, Manish, you got your answer? Hello?
Yes, sir. Thank you, sir.
Yeah.
Now it is clear to me.
Yeah.
Sir, other than food items, okay, if you want to apply this algorithm on clothes, so is it applicable for that?
Yeah. Yeah. Yes. Close. What purpose? What is your end goal?
Suppose we want to check the quality of the fabric.
Yes, you
So,
can do as well.
okay, fine, sir.
Because what
Yes, yes, sir.
was happening when you are uh, identifying a cloth, like if you want to identify silk and cotton. So if you want to differentiate silk and cotton, silk has a definite kind of spectral information. Cotton has its definite kind of spectral information. You can capture the hyperspectral image, and then you have to develop the algorithm, and then you can identify.
Yes, sir. Thank you, sir. It is really very interesting. Thank you very much for your guidance. Yes, sir.
Yeah. Any other questions? Hello.
So anybody else wants to ask any question?
Hello. Yeah.
So
I
one
think
of
I'm
the
disconnected.
participant, Dishri Zube Vincent, is asking about that any dataset which is available in public for the use.
At the moment, I think any data set, I'm not sure, but there are some remote sensing data is available. But remote sensing data is like hyperspectral imaging, but you have to some knowledge about GIS, Geographical Information System as well. If you want to play around, just go to ISRO, a website, and there is in ISRO website there are a lot of hyperspectral imaging data available. But again, he has to some knowledge of GIS as well. Then only he can process that remote sensing data.
Okay, sir. Anyone else? You all can ask questions if you have any, which may help you in your further studies or further research also. If you have any doubt in this field, then you may ask. Sir, good
Yeah,
afternoon.
Samir.
Sir, maybe a different
That's
question,
okay.
but
That's
I
okay.
feel, sir, actually, I'm also working in image
Yeah.
processing. But is it possible for you? Suppose I don't have the data set,
Mm-hmm.
and I have to prepare data set by own.
Yeah, yeah.
In this point, is it possible at this moment? Can you, if guide some points, so it would be great help for me as this at this time, if possible, sir. Thank you
And
so much.
yeah, so just
Maybe the answer may be not from at this appropriate at this time, but if sir, if you can, so I would be thankful to
s-
you.
yeah, so I have just one question from you. What kind of image processing you are using? Like, what is your end
Sir,
goal?
actually, it is a medical image processing,
Yeah, that's good.
and where I have to simply segment the image.
Oh,
But
segmentation is
yeah, but the image which is available in the
yeah,
public
yeah,
domain, the image is available.
yeah,
But for the problem which I want to finalized, so I don't have the answer. So I feel I have to go the unsupervised learning,
yeah,
perhaps.
yeah.
So I feel I'm able to clarify my problem at this moment.
PhD, always PhD studies is like that. So
Sir,
no
sir.
worries. That's very normal. So nothing
Yeah.
wrong in that. All good.
Okay.
So what I'm saying, if you
Yeah.
want to develop your own data sets, they do you know digital image processing in book?
Yeah.
So there are a lot of images freely available. You can download more than 1,000 images in the website on the website of digital image processing book,
Right.
and you can use that data for your initial segmentation, whatever approach. Like the cameraman image is most famous that we use initially for the segmentation to segment the camera man and the background. If you know the cameraman image or you don't know, I'm not sure.
Okay.
So there is a big data set available. And for the medical image data set, do you know Kaggle?
Yeah. Yes. Sure. Sure.
So Kaggle has lot of image data set available.
So
Just you have to explore.
there I
You
have
have to.
seen the dataset MIAS dataset actually is given there, but actually the part which I want to separate, the problem that I am facing, that suppose through particular algorithm I have separated the that the segmentation part has been done, but how to justify that the segmentation done is appropriate? I mean that,
Segmentation then is appropriate is the result output image. It will let you know
I mean,
like output.
in that image, in that area, actually, there is no particular division is given. So I myself is not confirmed whether the segmentation I have done is really giving the answer or not.
So what kind of thing you are like when you want to segment, there are two things you first decide in one, foreground and background. So which you consider as a foreground, which you consider as a background. That is clear to you or not?
Maybe not clear.
Okay, so foreground and background means, suppose in my potato case, the background is the like the labels, the back images, like, and I will show you that slide again. So I will explain you what I mean. So for my case, so the background is like here you can see these labels is the background. This top white is a background, and this is like
Your screen is not shared.
not shared. Okay, I have to share again. Just give me a moment. Yeah, can you now you can see Samir? Can you see it, Samir?
Yes, sir. Yes, sir. Yes, sir.
Yeah, so Samir, can you see this? These labels is the background,
Yeah.
and this black cardboards, you can say this is the background. This top white strip is the background, but this potato for me is a foreground. So that's my reason of interest. Like I'm interested in potatoes. That is foreground. The area in which
In
you
my
are in.
case, this is the
Yeah.
foreground. Yes.
So foreground is the area in which you are interested.
Yeah.
Background is the area that is you are not interested in. That means
Yes.
you don't want that area to include in your final output image. So
Yes.
So when I segmented, so I showed this, yeah. So you can see here, this strip, these labels, this black background, all gone. Just only potatoes were left. So all background is removed, only foreground. That means the potatoes are in front of me, nothing else. Everything else is 0 now. Only the data is in the potato area. So first you have to decide in your image is which is the foreground, which is the background. And then you have to decide, okay, so which kind of segmentation approach is applicable. There are many segmentation approach. Otsu algorithm,
Yeah.
canny age detection,
And upward.
thresholding,
Yeah.
lot of, it's a lot of, so you have to play around all different kind of algorithms. Then you have to decide which one is optimal in your case.
Definitely, sir. Definitely, sir.
So it's hit and trial thing. You can, nobody can give you a right away answer. Yeah, this is the best, and that's the PhD. You have to explore, fail, explore, fail, explore, fail, and at the end you will say, yes, this is the best. I got
Sir,
it.
may I have one request? If
Yeah, yeah, sure.
can you share your email ID if possible for further future contacts, sir, if possible.
Yeah,
Thank you so
yeah,
much. Thanks a lot, sir.
yeah, sure. So I will share email ID with, I think, Manish. You can ask from Manish Mathuria.
Thank you so much, sir. Thanks a lot, sir.
Yeah,
Yes, sir, I will share. Thank you,
yeah,
sir.
yeah. Any other question? Shailesh, Dr. Shailesh. Yes, Dr. Shailesh, can you unmute your microphone?
Sir, I was just replying to one of the questions actually, because Mr. Vikram, I think, had asked which are the other research areas in DIP.
Yeah,
So, like, I worked on a project which deals with tiles detection. Tiles can be
yeah.
detected according to various qualities, they can be classified.
Yes,
In
definitely.
Gujarat, we
Sure.
have a place called Morbi,
Yeah, I know.
where this tile, it's a ceramic tile hub. So there the
Yeah.
tiles are being manufactured and it is manually being done right now. So we have tried to implement that using image processing.
But tile detection, like you are using multispectral camera, I think, not hyperspectral.
Yeah.
Because hyperspectral is like it's a continuous wavelength, and multispectral camera is discrete wavelengths. So tile
Right.
detection, but
But it's,
in
yeah.
yeah, you can continue.
Yes.
So what happened, like when we start initially the research, it will start with the hyperspectral imaging, because the spectral resolution of the hyperspectral image is around from 1 nanometer to 10 nanometer, depends on your requirement. And then you will identify, like in your tile case, you can identify which wavelength is important, and according to that wavelength you will develop a multispectral camera, and then multispectral camera can be used further for, because like initially supposed 235 wavelength, from 235 you have to identify 10 wavelengths important for tiles detection, and for that 10 wavelengths you will develop another camera, multispectral camera, that is super cheap. Hyperspectral camera minimum cost is $40,000, but the multispectral camera is not more than $2,000. So it makes a big difference for the industrial application. And I don't know, in you are explaining like in the tile detection, so they are using the multispectral or hyperspectral. Do you know that?
We actually used bachelor series camera, high resolution. I'm not clear whether it is
So that means
multispectral
that
or not.
it means that is a RGB camera. It's not a high multispectral. Not a, it's a RGB camera at a very high resolution. It's not hyperspectral, not multispectral.
Right.
Yeah. Because RGB camera is, if at a high resolution, is it also get like more information. Yes, Manish Bhardwaj.
Yes, sir.
Yeah. So Manish, at the moment I don't have like hyperspectral images of lungs, because it's very hard to get them images. You have a live human beings. How can you get live human beings hyperspectral images?
So, but as technology has developed a lot, so I was thinking like, is there in some way to find out this thing?
You have to prepare your own data set, and you have to prepare, like, ask some volunteers. They are happy to say, yeah, I can you can scan my lungs for your experiment.
Okay,
It's a
sir.
very you have to apply for ethical issues. It's ethical, unethical. You
Yeah.
capture the images. So contact any medical hospitals, and then if you talk to a doctor or then to them, then they might help you to get some patients' hyperspectral images. Yeah, that might be possible. But why you want to get the hyperspectral images of lung? What is your end goal, basically?
Just to, sir, take the data irrespective of the in the response of COVID, and just to perform some research.
COVID hyperspectral data is like, if you ask about COVID, so yeah, it is possible, but again, it's very dangerous at the moment for COVID. So it's hard to identify COVID patient. If you identify them, then who is going to scan them? And then it's an interesting topic, but again, it's you need a ethical approval from the government, or there are some bodies that will approve you before you collect the data. So it's not an easy process, in short, but yeah, if you try, you have to go through a lot of steps, and then you are able to collect the hyperspectral images of lungs.
Okay, sir. Thank you very much, sir.
Okay, sir.
Okay.
It's a very good session.
Yeah, thank you.
And I am very thankful to you. Thank you for this very informative session. And I believe that this session definitely be useful for the participants and help them to proceed in the field of image processing. Those who are trying to work on this particular area, they will definitely get benefited.
Yep.
So thank you once again. Thanks a lot, sir.